One of the things that we talk about in this unit, that we talk about in photosynthesis and cell respiration, is oxidation and reduction, because they're happening all the time. And so I always like to talk about when these things are happening. So the best example I think that we could give of oxidation is when NADPH becomes NADP positive. Now there's also some electrons, there's also some hydrogen. But the idea is that oxidation is losing something. So that acronym, OIL-RIG, means oxidation is loss, reduction is gain. And so oxidation is when we lose something. In our case, like in our topics, you're always going to notice a hydrogen either be there or not be there. If something has hydrogen, I can oxidize it. If something doesn't have hydrogen, I'm not going to be able to oxidize it. It's already oxidized. Now, where does this happen? This got made in the first part of photosynthesis, and it gets used in the second part. So if I'd like to say where that happens, it happens in the stroma. Now, when we talk about cell respiration, we'll talk about these same processes and where in the mitochondria it happens. Reduction then would be the other way around, when NADP positive becomes NADPH, so I'm adding something, so reduction is gain. This happens on the thylakoids, and I would have also accepted granum or grana because that's a stack of thylakoids put together. Would anyone like to ask any questions about oxidation and reduction? So, I'll tell you, one of the things I like to ask when we're doing a quest, when we're doing a unit test is, does this happen in photosynthesis or cell respiration, or does it not happen anywhere? And I like to be sneaky. So I might ask you at some point in time, hey, when does that molecule get oxidized? And you would say to me, never, it's already oxidized. Or I might say to you, hey, when does this molecule get reduced? And you would say it doesn't because it's already reduced. So I really want you to recognize that when the H is there, it's already reduced. I can't reduce it more. When the H is not there, it's already oxidized. It can't be oxidized again. Does anyone want to ask any questions about that? Look for the hydrogen. If it's there, it's reduced. If it's not, it's oxidized. Now, question number 3. We need a bit more space, please. There we go. Question number 3 asks, what are the products of photolysis, which is the first part of photosynthesis? I on purpose called it by one of its other names. This is the light-dependent reaction, or the light reaction. So we're talking about part 1. The products are oxygen gas, NADPH, and ATP. Those are the products. Of those 3, these 2 are the ones that are used in the Calvin cycle, which is why they don't show up in the overall reaction for photosynthesis. Now for number 4, what is the main product of the Calvin cycle? The main product, because of course I'm using NADPH and ATP, so technically NADP and ADP are products of the Calvin cycle, but I want to focus on the molecule that's our carbohydrate. Specifically, I get 1 molecule of G3P, glyceraldehyde 3-phosphate, and every time the Calvin cycle happens twice, I can make a molecule of glucose. So, the product of the Calvin cycle is a carbohydrate. That's what we're getting at. 1 Calvin cycle gives me a small carbohydrate, G3P. It has 3 carbons. When the Calvin cycle happens twice, then I get my molecule of glucose that has 6 carbons. So on this screen, I'm going to mention 3 factors that affect the rate of photosynthesis. The word rate means how fast is it happening, and usually we would measure how fast it's happening by measuring how many products are being produced. So in real life, if I want to see how fast a reaction is happening, I need to look at how much or how many of the products are produced. Now, so that you know, I on purpose picked graphs that didn't have numbers on them because I don't want you to focus on numbers. I want you to focus on the overall pattern or the overall shape. The first factor is temperature. So just like temperature affects an enzyme, temperature will affect the rate of photosynthesis. Do you know why? Because photosynthesis uses enzymes. If the enzymes don't work, then photosynthesis cannot happen. So as temperature increases, the rate of photosynthesis increases to a maximum and then declines sharply. And that's because we know from previous lessons, enzymes become denatured or even coagulated if we increase the heat even further. So the idea is that temperature, just like it affects an enzyme, will affect the rate of photosynthesis, which is a bunch of reactions that require enzymes to function. So over here on the right, there is a graph. Along the bottom is temperature, and along the y-axis is rate of photosynthesis, and that's how all of the graphs are laid out. Rate of photosynthesis is always on the y-axis because it's what I'm measuring. Temperature, or whichever factor I'm trying to see the effect of, is always on the x-axis. I'm mentioning this so that when you go to make a graph in the lab that you're doing today, you remember that measuring how fast photosynthesis is happening goes on the y-axis. Now this is temperature. If you look, the line goes up until I hit a maximum point. And as soon as I go past that maximum, the rate of photosynthesis drastically and sharply declines. And that's because enzymes are proteins, and proteins become denatured or coagulated as heat increases. So if I were to ask you a question about this, I would very likely use a graph. I would very probably say, hey, here's a graph. Which factor am I talking about? Or which factor does this represent? Or perhaps I would say, hey, here's 4 graphs. Choose the one that represents how temperature affects the rate of photosynthesis. So you can absolutely expect a graph to show up. So as long as you recognize that it goes up and then straight back down, that is how temperature affects photosynthesis. Now the second one is light intensity. And don't confuse this with heat. A lot of times people confuse light and heat because if there is light, it generally feels like there's heat, but we're separating the two. As light intensity increases, so does the rate, but then it reaches a maximum and levels off. Now we're not talking about heat here, so I'm not talking about it's too hot and the enzymes can't function. The reason that as light intensity increases, maybe the rate of photosynthesis does not anymore, is that there are only so many molecules of chlorophyll. There's a limit to how much light can be absorbed at one time. So once all of the photosystems that contain chlorophyll have been activated, I can't do any more photosynthesis. I can only do as much photosynthesis as I have structures to accomplish the process. So light intensity increases the rate of photosynthesis to a maximum, and then it levels off. And the reason is the number of chlorophyll molecules that are available to absorb it is limited. So here's what the graph would look like. Just like the first one, rate of photosynthesis is on the y-axis, and the factor that I'm investigating is on the bottom. The word that this says is irradiance, which means how much light there is. So always the factor is on the x-axis, rate of photosynthesis on the y-axis. This section here is where light is actually having an effect on the rate of photosynthesis. So, to a certain point, increasing light intensity will also increase photosynthesis. So, on this particular graph, there's a portion where light intensity is affecting the rate of photosynthesis. But once I get over here, and the line is basically flat, light intensity is not changing the rate of photosynthesis anymore. So if I were to put a graph on a quest or on a test, I might ask you, hey, what would happen if I put the light intensity up there? And you would say nothing, because it's not making the rate of photosynthesis get any higher. So what I'm trying to convey is that sometimes when I ask you what will happen if I increase this factor, the answer is nothing. That's going to be a choice that's possible. Now the last factor is carbon dioxide. Carbon dioxide is required for photosynthesis. We know that because it's part of the Calvin cycle. As carbon dioxide levels increase, so does the rate of photosynthesis. It will reach a maximum and then level off, just like for light. Now the thing with carbon dioxide is that you don't need an incredibly high concentration of it, because if the concentration gets too high, other things can happen. So we only need a low concentration. That's my abbreviation for concentration there. Less than 5%, but I wouldn't expect you to memorize an actual number. And that's because if the concentration got very high, well then pH will drop. Carbon dioxide, when it's dissolved in a solution, can become an acid. That's the basic principle. And so if there's way too much carbon dioxide, this could cause enzymes to denature, just like it did with temperature. So if I were to continue the line on this graph down here, which shows rate of photosynthesis along the y-axis and carbon dioxide concentration along the x-axis, if I was to extrapolate and continue it on for a while, eventually it would drop off. The concentration has to get pretty high for this to happen, but in the long run, that's what could happen. So when you open up the lab, you'll see this is what's up there. This is what's on the screen. So to do this lab, you need to use the photosynthesis simulation located at that website. And I will go there in just a second and show you briefly how you use it. The sections that you need to complete, you'll see I put them all in bold so that you don't miss any of them. You need to tell me what the problem is, what the question you are trying to answer is. Now you'll see when we get to the site that you are going to test the effect of something on photosynthesis. And so I'd like you to phrase the problem, what is the effect of blank on blank? So really you just need to take that little sentence and fill in the two blanks. Now your hypothesis should be written as an if-then statement, just like we would have done for other labs where you had to make a prediction. So for example, in the macromolecules lab, you wrote if-then statements for macromolecule tests. If the Biuret test is added and turns purple, then it means a protein is present. So you're only going to write one statement because we're only investigating one problem. So you just have to have one statement. If you do something, then what should happen? So the first blank should always be, what are you going to change? In other words, this first spot should be your manipulated variable. The second spot should be what you think is going to happen, your responding variable. If you can fill that in, then you've already answered 2 of the variables in your lab. For the variables, I would like you to explain your choices. So rather than just say, color is my responding variable, explain why it is. So a manipulated variable is something that you, the experimenter, are changing on purpose to see what effect it has. The responding variable is always what you are measuring or observing. And controlled variables are something that stays the same in every trial or every test you do in your lab. The next thing that I'm asking you to do is to make a procedure. I'm not telling you exactly how you need to go about doing this. I'm going to give you some leeway in choosing, for example, how many times you're going to repeat it. I'm going to give you some leeway in which measurements you're actually going to use so that everyone's lab does not have to be identical. The thing I do want to mention, no step is too small. If you did something, you have to put it in the procedure. I should be able to give your procedure to someone who knows 0 about anything biology related, and they could follow the steps and do what you did. The steps should be numbered, so make a list, 1, 2, 3, 4, 5, 6, 7, 8, until you're done all of your steps. Your observations should always be in a table, and make sure that your table has titles, like if you're going to measure time, put time and maybe seconds or minutes as the unit. In your analysis, you need to make a Google Sheet file and make a graph, much like we did in our previous lab. So if you need help reminding yourself on how to make a graph, that would be great. Now, I indicated that you can make a separate sheet and submit it, and then I noticed that last time a whole bunch of you just copied and pasted your graph right in your lab, and so I'm just going to modify this instruction slightly. Could you please do that? It actually makes it easier for me to mark it when they're all in the same spot. It's relatively easy to copy and paste things, and if you'd like some help with that, I would be happy to show you how to do that. I gave you some instructions. For example, graphs need a title for the graph, and for each axis you need to indicate which units there are, and you need to give consistent divisions on each axis, which is really easy when you use Google Sheets. It does that for you. In your conclusion, you tell me what your hypothesis was, and you accept or reject it, and you give some reason why. Based on my data, the hypothesis is accepted because, or based on my data, the hypothesis is rejected because. Evaluation always has to have 2 things. Any 2 things that we could do if we were to do the lab again that would make it better. These could be things that need to get added. So if you think maybe something was missing on the simulation, or it would have been better if there were more choices, that's the kind of thing that you need to put in your evaluation.